from acute liver disease. Next, we use our experimental models to prove this and to try to find out the mechanism. Biochemical, we used C57 black cis mice which are sensitive to cone hepatitis, and on the same background galectin-3 knockout mice. Biochemical analysis showed that galectin-3 knockout mice, is in red, after cone treatment has significantly lower levels of IST and ILT. Also, when you compare the livers 24 hours after disease, you can see massive coagulative necrosis on the field of C57 black cis wild type mice, while galectin-3 knockout mice has only focal necrosis. When we focused our attention on liver sinusoids, we found out that there was significantly infiltration in the livers of wild type mice compared to healthy animals. Then we calculate all these necrotic fields and find out that percentage of total necrotic liver tissue was significantly higher in wild type animals compared to galectin-3 knockout animals. So, first conclusion, target disruption of galectin-3 gene protected from cone liver injury. Then we focused our attention on inflammation. We found out that galectin-3 knockout mice has significantly low number of pro-inflammatory cytokines, TNF-alpha, interferon gamma, interleukin 17, interleukin 4. And then we tried to find out if this happens also in vitro, if there is a problem in these cells in vitro after cone stimulation. We find out that this function only in vivo systems. So in vivo cone hepatitis is important for all this disruption of cytokines. Then we also focused in the liver. We found out that there was significantly low number of activated lymphoid and dendritic cells in the liver of galectin-3 knockout mice, including all these effector cells, CD4 T cells, macrophages, NK T cells, dendritic cells, and so on. And we find out by intercellular staining that these cells produce less amount of TNF-alpha, interferon gamma, and importantly, much more interleukin 10, which is protective in this disease. Then we also investigate, I will go through these slides very quickly, investigate all this population of immune-mediated cells, NK T cells, dendritic cells, and find out the same, that galectin-3 deletion significantly reduce influx of cytokine-producing NK T, and dendritic cells. Next, very important is that galectin-3 is important not only for specific and also for innate immunity, because deletion of galectin-3 led to alternative activation of macrophages. Macrophages, which had markers of alternative activation CD206 and 273 and produced interleukin 10, was significantly higher present in the livers of galectin-3 knockout mice. Galectin-3 has pro-inflammatory role in this disease on one hand, and on the other hand it is important for apoptosis. In cone hepatitis, cells are activated by cone A on one hand, but on the other hand they also die after cone induction by apoptosis. We find out that galectin-3 deletion enhanced apoptosis of liver infiltrating mononuclear cells, both in the liver and in the spleen. So, galectin-3 is important in this disease. But what is galectin-3? Why this is all important for the therapy? We then, in our collaboration with friends from Lund University in Sweden, we presented them our results, and they managed to make selective inhibitor of galectin-3. This selective inhibitor of galectin-3 is TD139 galectin-3 inhibitor, and we tested it in this model. We gave inhibitor of galectin-3  2 hours before cone A and immediately after cone A, and we find out that this inhibitor really did its job. Galectin-3 inhibitor prevents cone A hepatitis. As you can see, biochemical analysis show significantly decreasing both IST and ILT, and livers of these animals were almost as control animals. Galectin-3 inhibitor managed to downregulate pro-inflammatory cytokines, interferon gamma, interleukin 17, TNF, interleukin CD4, and significant increased presence of interleukin-10, which has protective role. When we analyzed all these inflammatory cells, we find out that galectin-3 inhibitor acts primarily on T cells, CD4 T cells, cells which produce interferon gamma, interleukin-17, interleukin-4, so all cytokines, but manage to produce much more much more influx of CD4 positive, interleukin-10 positive cells, which are protective in this disease. Not only T cells were affected by inhibitor, macrophages also. Macrophages, so galectin-3 inhibitor favor alternative activation of macrophages and reduce apoptosis in the liver. So our second and last conclusion was galectin-3 has pro-inflammatory role, galectin-3 deficiency prevents connect-induced hepatitis in mice, and galectin-3 inhibitor could be tested as new potential therapeutic agent in this disease. These papers, this work was published last year in 2 major hepatology journals. So that's all. Thank you for your attention.
Thank you very much. Questions?
Okay, I have 2 questions. Actually, the first one is the most important one. We have in the interleukin-33, there are 2 major pathways. The first one is suppressing the apoptotic pathways, which is something with the apoptotic, pro and anti-apoptotic pathway, which is clear. But for the first one, involvement of different cells, blood cells, which are entering the hepatocytes, I'm not quite convinced by that one, in spite of the publication. If I am the reviewer, I will ask you to do the migration essay in cell culture.
Okay, in this disease, cells are polyclonal activated in the whole organism of the mice. When you inject CoN-A, all cells in the organism were activated, both in the circulation, both in the spleen, and then during first 2 hours they migrate in the liver. I did not present here, but we did as supplemented figures that in the spleen, the situation is diverse. Opposite. Yeah, opposite. So it was some indirect conclusion that cells migrate in the liver.
Okay, thank you. Some more questions? I have small questions. Actually, it's completely not understandable for me, many things. But anyway, if you think about modeling, I see you are doing gene expression, right?
Yes.
So that means you don't have concentration of the proteins, you actually have some activation. So you don't have quantity of the numbers, you have something is going up or down, right? In terms of gene expression.
No, you have control is 100% and
okay.
What is above.
But that's not concentration or is?
No, you measured concentration also when you did the real-time PCR also.
So you have numbers?
For genes, you asked me for genes,
yes.
PCR, yes.
Okay, so, okay, but as I understand, we can do maybe some kind of statistical analysis and data mining, but I'm not sure for this modeling process of kinetics. I don't know, that will be interesting.
Let me ask the same question another way. Did you perform any analysis on the protein level?
Yes, Western blot.
This is the most important question.
Western blot, yeah, Western blot.
You get the numbers from that.
Yeah, okay, Western blot analysis, okay.
Yeah. Because
I'm not familiar in
yeah, if you have that numbers,
yes,
we can do a modeling. Okay, thank you again very much.